I just want to thank the organizers, Shigui and Zara, for the kind invitation to speak at this nice event. My name is Yurij Salmaniw. I'm actually a PhD student at the University of Alberta. And today I'm going to talk about modeling habitat loss with partial differential equations. And this is joint work with my two supervisors, Dr. Hao Wang and Zhangwei Shen. So I thought I would just start with some motivation. So in 2001, the late Steve Irwin was asked what he saw as the biggest threat to Australia. And he said, the biggest issue to Australia at the time. And he said, I believe our biggest issue is the same biggest issue that the whole world is facing, and that's habitat destruction. So there's a picture of Steve Irwin. I used to watch him when I was younger. But more seriously, habitat loss has many negative implications with many cascading effects. And so some things include reduced pollination and seed dispersal. So many plants rely on, say, insects or small mammals to spread their seeds around. And if those animals disappear, they will struggle to propagate. Reduced climate regulation. So for example, roots in the forest will help stabilize things for events like floods. And so if the forests disappear, floods can be more likely. Logging can increase likelihood of forest fires, stuff like that. Reduced pest and disease control. So there's some, maybe you've seen some news articles about our favorite banana, and we've sort of homogenized the banana population. And when that happens, the pests and diseases can spread more easily. And so that's a current issue with the banana that we see in the grocery store today. And there are a lot of other indirect effects. So just decreased food security, things like coffee. We all like coffee, or most of us probably like coffee. Decreased ecosystem health. So this is kind of a big problem. And all of these effects are primarily driven by a loss of biodiversity. And so if the reduction or extinction of species is bad, and habitat loss is a primary driver of species loss worldwide, then we must clearly understand how and to what extent habitat loss actually does this. So again, we know that habitat loss is the primary driver, but some of the details inside of that can be confusing. And so, of course, mathematical modeling offers a method apart from costly data collection and issues with timescales. So for example, we do have a lot of data on habitat loss, and even though it's getting cheaper these days to monitor some of these things, it still takes a lot of time and effort to actually go and collect all of this data, match the data to things like rates of habitat removal and the fragmentation of the habitat. And then there are additional issues with timescales. So if a disingenuous logging company comes in and says, we want to log this forest, we've done all of these studies and we recorded things for 10 years and we saw that there was no reduction in local species, so everything is fine. But if it takes longer than their recording time to actually see the realized effects of habitat loss, then that wouldn't be a very good observation at all. And so maybe modeling can help us get at some of those more complex issues. So if we're going to model habitat loss, we need to define habitat loss in a meaningful way. And it turns out that it's a little bit complicated in the modeling and the ecological literature. So I'd like to show this little comic. It's a situation here where we have 14 competing standards. Somebody shows up and says, 14, that's a ridiculous amount. We need to come up with one single universal standard that covers everyone's cases. And then soon after, there are just 15 competing standards. So I'm not trying to specially define or redefine habitat loss in any way, but I think it's important to be clear about what exactly we mean by habitat loss, because if we want to study this empirically or mathematically, and everybody's working with different definitions, and they say habitat loss causes this, then someone else says habitat loss causes this instead, and they're talking about different things, then clearly there's reason for maybe contradictions to arise. And so just to kind of get at this a little bit more clearly, a common definition And by common, I guess I mean if you Google habitat loss or habitat destruction and you click on the first or second page, you get this definition. It says when a natural habitat such as a forest or wetland is altered so dramatically that it no longer supports the species it originally sustained, plant and animal populations are destroyed or displaced, leading to a loss of biodiversity. And then it says, note, habitat destruction is taken as synonymous with habitat loss. And so if we take that definition, it maybe ignores other things like habitat degradation. So what if the habitat is only decreased in quality instead of completely removed or destroyed? And then what about things like fragmentation? So how the landscape is fragmented and broken up as we slowly remove habitat. So again, this definition appears on a web page, but if you go to the primary source, you have this book, Conservation Biology for All. And so furthermore, for modeling, this is a little insufficient because we need to try to define habitat loss a priori so we can actually make predictions about the future impacts of the lost habitat. And so how do we usually model habitat loss, or how has, what's the approach that many models have taken up until now? It looks something like this. You have some portion of available habitat that is removed. So you have, say, total habitat of 1, and you remove some proportion of it. That decreases the carrying capacity of the system, and then the species feel negative effects through the decreased carrying capacity, possibly leading to deterministic extinction. So in particular, this is pretty much describing most of the ODE models. So anybody who's familiar with this area, maybe they know Tillman did some interesting models in the 1990s, and he used this construction. But I think that this maybe cannot capture all of the relevant information. And why is that? There's some reasonable inquiries one might make. What if the habitat isn't instantly removed or destroyed? So possible intermediate effects. That's getting at the idea of degradation. What if the species aren't impacted by changes equally? So again, maybe the habitat is getting degraded, but it actually depends on the species as well, how much that degradation will affect them. Questions about the configuration of habitat, how does that impact the results or the outcomes? And then maybe combined nonlinear effects of all of the above. And so again, if we don't have some sort of spatially explicit factors included, then it's hard to get at many of those properties or questions above. And so I think that a partial differential equation framework provides a nice additional perspective that is spatially explicit and can actually get at the heart of some of these issues above. So we'll start with the first project that I've been working on. This is for habitat degradation. And so a rough definition, entire habitat remains intact, but the quality is degraded. So you have the habitat, it's still there, and the species can still, exist in that poorer habitat, but perhaps the quality in select areas is degraded. So this includes things like agricultural development, extraction of natural resources, such as selective logging, pollution, and climate change. So all of these things can affect the habitat a little bit, but not completely remove it. And so what we want to do is use the reaction term in a reaction diffusion equation setting to model the dynamics when a portion of a total habitat, which we denote by omega, has been degraded. So omega is a smooth bounded domain. We're just working in bounded domains. And for most of this, I'm thinking in maybe 2 dimensions, but of course, 3 dimensions for aquatic creatures could also be relevant. So what we do is we partition the habitat into 2 disjoint pieces. So we want it such that these sets G and B, when you put them together, you get back the entire habitat. Capital G will denote the undisturbed habitat, and so G for good habitat. And capital B is the degraded habitat at some level. And so B would be for the bad habitat. So we can visualize this in 2 spatial dimensions. So we can cut up the habitat pretty much in any way that we want. We don't have any major restrictions or any restriction at all really on the set B. And so it could be pieces that are interior to the set G. It could be that B makes up most of the habitat if it's really bad. It can touch the boundary, things like that. So there's no restrictions on that set B. And so then what we do in a competition model setting is we assume that the reaction terms are of the form here. So like a Lotka-Volterra competition with all parameters equal to 1, so as to isolate the effects of the habitat loss. And so in the region G, it's sort of business as usual, but then we assume that the reaction terms are now just exponential decay in the region B. And so we just have decay for U and decay for V at some rate C1 and C2 that will be greater than or equal to 0. And so since we're working in these separate sets, G and B, it's convenient to introduce the indicator function where this 1 sub k is equal to 1 if x belongs to that set k, and then you're 0 otherwise. And so this allows us to write the model in a way that's maybe more familiar for analysis. So our two species competition model now takes the following form. We've got some diffusion at rates D1 and D2. Because of the symmetry of the system, we're always going to assume that D1 is less than D2. So we always assume that species U is the slower diffuser. And then when we simplify our reaction terms, we can actually write it in this way. So if we look, we have these MC1 and MC2 terms, and then we have the competition terms on the right here. And so if you fill in this MC1, which is just equal to the indicator function of the set G minus Ci times the indicator function of the set B, if you simplify this, you pull out the whole competition term restricted to the set G, and then you have just decay in the set B. So this is just, again, writing it in a little bit more of a succinct way. And so what our goal was to analyze the stability of the steady states depending on these diffusion rates Di and the level of impact Ci. And so again, this is a degradation model where the parameter Ci is somehow measuring the level of impact on the species U and V. And so this is kind of a parameter that's specific to the species, whereas the sets G and B kind of denote the arrangement of the destroyed habitat. And so this is a theorem that we found that was recently published in the Journal of Mathematical Biology last February, I guess. And so if we fix all parameters ordered as I stated earlier, 0, less than D1, less than D2, and we assume that C1, the level of impact on the slower diffuser, is just fixed and positive, then for any non-trivial data, there exist some critical thresholds, the C2 underline star and C2 overline star, which are ordered and they're smaller than C1, such that we have the following three regimes. So if the level of impact C2 on the second species V is too large, so if it's in the interval C2 overline star to infinity, then it turns out that the slower diffuser will always win. So the slower diffuser will always drive the faster diffuser to extinction as T goes to infinity. On the other hand, if the impact of the destroyed or degraded environment on species V, this parameter C2, is intermediate, so it's between C2 underline star and C2 overline star, then there's a unique coexistent state that is asymptotically stable. So we converge to that as T goes to infinity. And then finally, if C2 is small enough, so if it's between 0 and this lower threshold C2 underline star, then we have that the faster diffuser will actually displace the slower diffuser as T goes to infinity. So in particular, we have a limiting case where all the parameters are fixed as stated, but C2 is actually 0, then we have that the faster diffuser will always displace the slower diffuser. And this is as long as the region B has positive area. So I said that the region B has very limited restriction. The only real restriction is that it has positive area somewhere. So in the sense of the Lebesgue measure. So you can have some like fractally sets if you want, but it has to have a positive measure for this result to hold. So that's a complete description of the global dynamics, and we can visualize this in a little region plot. And so what we do is we fix the parameters C1 and D1, so the level of impact on species U given by C1, and the diffusion rate of species U given by D1. We just fix those equal to 1. And we look at the parameter space C2 on the x-axis and D2 on the y-axis. And so again, D2 is greater than D1, so we start our y-axis at the value 1. And then C2 is between, well, 0 and 1 to get between 0 and C1. And then we have greater than C1 on the right here. So if we look on the right side of the panel, species U is the winner and will always displace species V. And this is pretty much a classical result. Again, the slower diffuser always wins. But then we have this regime here where even if C2 is a little bit smaller than C1, species U will still displace the faster diffuser. But then, as we keep going, again, for some D2 fixed, we have this window where a coexistent state appears. So we have U tilde V tilde being globally asymptotically stable. And then again, as we continue to decrease this parameter C2, we have that the species V is now the winner. So we have this interesting kind of change of dynamics as this parameter C2 varies. So that was the main result for the degradation case. And now I wanted to move into a habitat destruction model. And so we're going to connect this to the degradation model, but we'll kind of start with the formulation for the destruction problem first. And so a definition of habitat destruction occurs when a natural habitat is altered so dramatically that it no longer supports the species it originally sustained. So this is actually closer to the definition given at the very start that seemed a little bit insufficient if we just call all habitat loss as habitat destruction. So instead, I want to restrict that or save that definition for habitat destruction itself and have habitat loss as kind of an umbrella term for all of these different forms that it can take. And so examples include land conversion, so such as, again, agriculture and maybe urbanization, clear-cut logging, so heavy deforestation, heavy pollution, and forest fires can all be considered as habitat destruction. And this kind of gets at the point that habitat destruction is perhaps related to more extreme forms of habitat degradation. So I don't think that habitat degradation and destruction are so much a binary classification as much as maybe a spectrum where a little bit of habitat degradation might be fine for some species, but it might actually be destroying the habitat for other species. So it should be species dependent. And so we still split the habitat into disjoint regions, G and B, the good habitat and the bad habitat. But instead of assuming decay at some rate, we will now make the kind of limiting assumption that the region B is immediately lethal to the species under consideration. And so because of this, we actually have to introduce a much stronger condition on that set B. And so we assume that B is compactly contained in omega. So we have some distance between the set B and the boundary of the entire set omega. We assume that the boundary of these sets B are smooth, and we assume that it's made up of finitely many subregions, each of which are simply connected. So that might be a lot for an assumption, just as written. So I've got a little picture here. Excuse me for a second here. Sorry about that. Yeah, so on the left here, we have a set of allowable regions. So we can have any number of, or any finite number of disjoint regions B here. Each of these are simply connected and they have relatively smooth boundary. Whereas on the right, we have a few configurations that we are not allowing. So this set here, we have a hole that appears, and so we don't consider those cases. On this set here and this set here, we split the domain, or we touch the boundary of the domain. So we need some space there. And in this case, we actually touch the boundary and we cut the domain away. And so just to clarify one piece here, when we introduce holes, as we'll see in a second when the model is introduced, we can actually consider these cases. It just involves a little bit more kind of technical explanation of how to treat those cases. So this isn't necessarily ruled out, but for simplicity, we just ignore these cases for now. The touching the boundary cases are a little bit more challenging, but I think or challenging mathematically, but for biological application, I think this isn't such a big deal. So in this context, our scalar equation takes the following form. So we have, again, a reaction diffusion equation. This time we've just written a general function f of x and w on the right-hand side. And again, we write omega set minus b to denote the remaining habitat, the good habitat. We take a homogeneous Neumann boundary condition on the outer boundary of omega, and then we assume a homogeneous Dirichlet boundary condition on the inner boundaries along the boundary of B. So this is now a habitat destruction problem with destruction confined to the region B. And so again, if we go back with this in context here, if we had a set B with a hole, essentially all you do is treat it as two separate problems. And so you would consider the problem in the set omega take away B, and you would have your zero Dirichlet boundary condition along the outer set or outer boundary of the interior set B. But then you would have this interior set here, which also has a Dirichlet boundary condition. But then that problem there would actually just be a classic Dirichlet problem with a homogeneous boundary condition there. And so you could treat that as two separate problems and then just look at the eigenvalues of each. And so our goal is to establish connections, if any. Spoiler, there will, of course, be connections between this degradation problem introduced previously and this destruction problem I've introduced here. And so in order to establish a useful connection here, we have to go back to the degradation problem. And in particular, we want to go back to the scalar degradation problem as a starting point. So the equation for the degradation model is essentially the same when we generalize it a bit. So again, we have our reaction or our diffusion occurring here for species W. We have the business as usual in the region G. So again, G is the remaining habitat, the good habitat, and we just have our whatever form this would be. It's going to be roughly of logistic shape. We want it to be concave down in the variable W. And then we have our decay at rate C confined to the region B. And so in order to actually establish a connection between these two formulations, we ask the question, what happens in the limit as C goes to infinity? So again, we have this degradation in the region B. And if we take arbitrarily large levels of habitat degradation, what happens to the solutions of this problem? And so we have a theorem. First, we assume that this parameter mu1 comma infinity is not equal to 0. So I'll come back to this in a minute. I didn't want to bog the talk down with too many technical details. But it's just this parameter, an eigenvalue we want to be non-zero. And then if we denote by W sub C and W infinity the unique solutions to the degradation and destruction problems respectively. So again, W sub C is going to be the solution to the degradation problem depending on this parameter C. And then W infinity, which is going to be the candidate limit for this problem, is actually the solution to this destruction problem here. So if we assume that the initial data satisfies some technical assumptions here, which again, I'll talk about in more detail, they have to belong to the set C sub B1, and we want the support to be contained inside the remaining habitat. So under these assumptions, we actually have that in the limit as C goes to infinity, this W sub C converges to W infinity uniformly in space and time. And so I should try to be careful here in the first theorem for the degradation problem, I was talking about convergence as time goes to infinity. So as we take t to infinity, it's converging to some steady state. This time, I'm actually considering the convergence in the parameter c. And so we actually want to obtain convergence in space, but as well as time. And it turns out that this convergence as c goes to infinity holds uniformly across all space and time. So for example, as t gets larger, it's not like things kind of escape as you take c larger. So you can just take this limit c and it holds uniformly across the entire space-time domain. And so again, we have a little visual depiction of this problem. I guess I said that this was, I guess for application, it would be in 2 and maybe 3 spatial dimensions, but this convergence result also actually holds in any spatial dimension. And so here's an image depicting the steady state solution profiles as c varies. So of course, this was for the time-dependent problem in omega cross 0 infinity, but it also holds for the steady states. And so this is a picture of the steady states as they depend on the parameter c. So again, these different lines here, the different colored lines going down, are the solutions w sub c. And so blue is c equals 1, and we have c equals 2 for the orangey red, c equals 10 for the yellow. And then in the black line, we have the limiting solution as c goes to infinity. And so we see that it monotonically decreases and it converges to 0 in that set B. And then you have this solution that is kind of like a Neumann-Dirichlet mixed boundary condition on each endpoint in the remaining regions of habitat. So I have some quick comments for that theorem. As I said, mu1 comma infinity is a principal eigenvalue which determines the stability of the trivial steady state for the limiting problem. So if this is 0, we still have convergence, but it's just a little bit more complicated to look at. So we've left that out for now. We use a standard concavity condition on the reaction term. And so we needed this to get some uniform control for all time as c goes to infinity. And so this may hold under the weaker condition of subhomogeneity, but when you assume mere subhomogeneity, we don't actually have an explicit control of the rate of convergence in time. So it may, so under concavity, it's going to converge exponentially in time. But under the condition of mere subhomogeneity, the rate of convergence in time might be much slower. And so we need a more explicit control. The set C sub B1 are just differentiable functions that vanish along the boundary of the set B. So you can think of them, this function space as C01, but instead of 0 on the outer boundary, we're now constructing a set which has zeros along the inner boundary. And the condition on the initial data is mostly a technical assumption. So again, we need some control near the boundaries. And so we need some space between that set B and the interior or the viable habitat omega takeaway B. And so then we have some other useful or interesting results that came out of this that I'll just share in words. So we have principal eigenvalues corresponding to the degradation problem and the destruction problem. And it turns out that these eigenvalues converge monotonically. And so as you take the parameter c larger and larger, these eigenvalues are increasing to some maximum value. The eigenfunctions also converge strongly in a suitable space, but that convergence is not monotone, or we at least couldn't show it to be monotone, but I don't expect it to be. And then we actually show a similar convergence result in a Lotka-Volterra competition system in that same limit. And in this limit, coexistence is no longer possible. So we recover the slower diffuser always wins result. And so this is kind of interesting from the habitat loss goal, because under habitat degradation and varying levels of impact due to the degradation on 2 competing species, we can actually facilitate coexistence. So we can have a trade-off from the fast and slow diffusion with somewhat higher resilience from the habitat degradation. And then in the habitat destruction case, it turns out that the slower diffuser always wins every time, and there's no possibility of coexistence. And so this is consistent with what we actually see empirically, in that as we destroy habitat, we decrease biodiversity, and there's always some losers in that game. And then we have only a few species that remain afterwards. And then we found that the scalar solutions are monotone in C, so they're monotonically decreasing in C, which is expected, but the solutions to the competition system may not be. So you end up with this trade-off where C is causing the system to decrease, but then as the solutions decrease, the competition between the two species actually decreases as well. And so it's not easy to obtain a monotonicity result in that case. So now, just quickly, I will talk a bit about fragmentation and some recent directions that I've been working on with my supervisors. And so habitat fragmentation is usually described as the breaking apart of continuous habitat into distinct pieces. And so this includes urbanization, agricultural development, deforestation, pretty much any form of habitat destruction or habitat degradation kind of brings fragmentation along for the ride, because we end up splitting the habitat into these more separate and isolated regions. And this is, again, the definition I gave above is kind of a standard one, but it's a little bit difficult to use for quantitative study of fragmentation, because it's quite vague. It just says the breaking apart of continuous habitat. And we want to try to come up with a way to describe fragmentation without having to appeal to such a vague definition. So we kind of want some measure to describe how the landscape is broken up into these different pieces, and especially how that breaking up might affect the species in that habitat. So there are many challenges when studying fragmentation quantitatively. Empirically, it's difficult to study because at least the existing studies we have, they yield contradictory results. And so they try to roughly measure fragmentation using a number of existing measures. And then they look at species abundance, and they will just compare, say across a few years, how the abundance changes with respect to this fragmentation. And some studies say that the fragmentation decreases the biodiversity and is a net negative. But then there are some studies that show that it might be positive, even though if the change is very weak. So it's not entirely clear what's happening in those cases. And it's difficult to separate the process of degradation and destruction from fragmentation. So in practice, we might want to isolate these three effects and say, what is the most impactful? What should we focus on to prevent or mitigate the most? But in reality, these processes all kind of happen at the same time. And so that makes it even more difficult to study empirically. Furthermore, when you're studying it, we need to ask the question, what is the correct metric to consider for fragmentation? So questions come up like, fragmentation, should it be measured independent of habitat abundance? And so, for example, some of the metrics that exist to measure habitat fragmentation depend heavily on habitat abundance, and they've depended so heavily on habitat abundance that the values that it actually outputs as the measure may not be super meaningful. There's questions about when or how to normalize. So if you just have a measure between 0 and infinity, depending on how that measure scales, it might not have a lot of meaning to say, this is a 1 level of fragmentation, and this is 100 level of fragmentation, and this is 1,000 level of fragmentation. And the numerous metrics that we have are not consistent in this way. So some are normalized, some are not normalized, and that increases the difficulty of interpreting those results. And perhaps most importantly, we want to know how to keep these measures connected to the ecology. And so many of the measures that exist are almost from the perspective of geometry and shape analysis. And so it's just talking about the properties of these different shapes of the fragmented patterns. But in some ways, that might be so removed from the ecology that even if you can describe the fragmentation using a certain measure, it's not obvious that that will actually be connected to the ecology itself. And so all existing efforts, and when I say all, at least from the review papers that I've found that have gone through many fragmentation measures, a lot of them can do some of the above, but they can't do all of it, or they can't address all of the issues that get raised. And in particular, that last point, how to keep it connected to the ecology itself. Many of the papers just state it's not easy to do that, and they don't have a measure that they know of right now that does so. So what we want to do is take motivation from the previous efforts that I've described to try to construct a new metric that is perhaps more meaningful, or at least a different perspective to the existing ones. And so to do so, we're going to use the following eigenvalue problem. And this problem actually comes up in studying the stability of the steady states of the previously introduced habitat destruction problem. So it's an eigenvalue problem with a sign indefinite weight, m of x. We have a homogeneous Neumann boundary condition on the outer boundary, and then we have a homogeneous Dirichlet boundary condition on the inner boundary. So as I said, closely related to a linearization procedure. And this function m of x is going to describe the habitat quality in the remaining region, omega take away b. And so some of the current progress, well, we have the existence of a principal eigenvalue lambda 1 depending on the function m and the region b. We have some monotonicity results with respect to resources and subsets. So if there are bigger resources, or if there are more resources, the eigenvalue gets smaller. And if the set B in the sense of subsets is larger, then the eigenvalue also gets larger. And this is consistent with what we would want a measure of fragmentation to do. This eigenvalue is continuous with respect to changes in the set B, and changes we measure in the sense of the Hausdorff distance. So again, a reasonable expectation for some measure of fragmentation. If you change the habitat a little bit, you hope that the measure only changes a little bit as well. The eigenvalue goes to 0 as the measure of the set goes to 0 in an appropriate sense. And so this means that as the destroyed region goes to 0 in measure, the eigenvalue goes to 0, which is good news for the local species. But then there are also a sequence of habitats B sub n, so that the eigenvalue goes to infinity as that sequence of habitats goes to infinity. And so again, this is kind of chopping up the landscape in a pathological way. So this sort of shows the limits of this as a measure of fragmentation. And then we also have some analytical results for simplified configurations, but those maybe aren't as interesting biologically because they're so far removed from what the fragmentation patterns look like in real life. And so preliminary results show that this eigenvalue may be an appropriate proxy for fragmentation. And so for fragmentation per se, which is to say fragmentation independent of habitat amount, all we need to do is hold the volume of that set B fixed, and then we can consider the impact of fragmentation alone. And so if you change around the geometry of the set B, this is going to change the size of this principal eigenvalue. But if we restrict the volume of the set B, this is getting at something depending only on the fragmentation pattern and not on abundance. So this gets at some of those previous questions related to existing fragmentation measures where they can't really make this distinction between habitat abundance and the actual pattern of the habitat. And so our goal is to use this to create a novel measure of fragmentation that is normalized and remains connected to the ecological or biological processes. And so without getting into too many of the details, it's normalized between 0 and 1. that's going from the most to the least fragmented. So 1 would be at the kind of the ideal case where the landscape is completely connected or the destroyed region is completely connected. And then going down to 0 would be like taking the pathological constructions of those sets Bn and sending lambda 1 to infinity. It turns out that the measure is scale independent. And so as you change the, say if you take a rectangle of some size and you blow it up, it's actually going to give you the same measure. So again, it's really getting at something to do with fragmentation. And then it remains connected to ecology because the measure comes directly from this eigenvalue, excuse me, directly from this eigenvalue lambda 1. And excuse me, comes directly from the value lambda 1. And lambda 1 is coming directly from this explicit assumption on the movement behavior of the species. So it's directly related to diffusive movement. That's due to the Laplacian term, of course. And so this also hints at how we might be able to use this measure to construct additional measures that if the species is found not to follow roughly diffusive or random movement, then we could come up with a different eigenvalue that includes the different movement mechanisms to come up with alternative measures that again remains connected to the species. Okay, so I think I'm just about out of time. I'll have a quick discussion page here. We talked about 3 projects for 3 forms of habitat loss. There's degradation, in which we found 3 regimes in a competition system, and we have a complete description of those global dynamics. Coexistence was facilitated by differing impacts of habitat loss. So we had that window of parameters where there's coexistence as a possibility. We have a destruction problem, and we establish a rigorous mathematical connection between the degradation and destruction problems. This is both for the scalar problem as well as the Lotka-Volterra competition system. And we find that in the limit, coexistence is no longer a possibility. So one species always wipes out the other. And this is, again, consistent with what we see in existing habitat loss studies, where habitat destruction decreases biodiversity. And then we talked a bit about fragmentation and how we're trying to develop a more robust quantitative measure of fragmentation. We're using the tools from the study of the dynamics to construct this measure. And so this principal eigenvalue seems to be a reasonable proxy, satisfies some reasonable properties that are consistent with how we would want to measure to act. And this is kind of our starting point. And so hoping that I'll get to see some of you in a future talk where I can go into more detail about that fragmentation measure. And so, yeah, thank you all for listening. Thank you to the University of Alberta and to, of course, NSERC for their funding.